Hey, it's George from Racks for Reptiles. I'm going to do a wee video here on genetics, how genes work and how to work out your odds when you're breeding. So we'll take it right back to the start. This funny looking shape here is just to represent a strand of DNA. Now, DNA is basically information that your body uses to create proteins. Each section of a strand of DNA can be known as a gene, and that's basically a stretch of DNA that codes for traits. So for example, this section here might code for eye colour or skin pattern or something like that. It's basically just a set of instructions to make proteins, and proteins are basically the building blocks that create phenotypes, and phenotypes are like your colour patterns, your mutations, your morphs. So let's have a look at this shape. This shape here is a chromosome. When you're breeding ball pythons, they have 18 pairs, so that's 36 chromosomes in total. All this is DNA compressed into one chromosome, and same on the other side, one from the mum, one from the dad. Simple as that. These areas marked here, these are known as the locus. So this is the area where the gene is located. You've got a pair of alleles at each section. This is basically different forms of the same gene. So this is why people say you need 2 copies of one gene to make a super or to make a recessive visual. So for instance here, I've marked that as pied. This has 2 copies of the pied gene, so this would be a visual pied. If it only had the 1 copy, this would be a het pied, because 1 copy doesn't show visual. So here, this only has 1 copy of the pastel gene, and the other side is normal. So this would be a single gene pastel. If it had the 2, it would be a super pastel. And then the same again with the spider. So now that you know we have to have 2 copies to make a super, or 2 copies to make a visual recessive, the next stage is finding out how we work out the odds of what you're going to be breeding. You've probably seen this before. This is what's called a Punnett square. So I'll keep this in there. On the Punnett square, we'll take for example the spider. What we're going to be doing is breeding a spider to a normal. So spider has 1 copy of the spider gene. Mark that at the top box. 1 copy of the normal gene, marked here. The normal would have 2 copies of the normal gene, so put that at the side. And all you're doing here is filling out the box: spider, normal, 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 spider, normal, 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 here. So as you'll see here, there's 2 boxes filled out that have a spider gene, 1 copy, 2 that has normal, so that gives you a 50% chance of hitting the spider gene and breeding spider to normal. So moving up from that, if you were to breed a spider pastel to a normal, so that's 2 genes to a normal, it's the same again. The normal There's two ends. So that's the normals down the side. The spider gene and pastel gene, this works a bit different. So the spider gene, take them by allele. So, for example, spider has a spider and a normal. S for spider, N for normal. Pastel has a pastel and a normal. So you'll mark it like that. Take the first letter from the spider, first letter from the pastel, put it here. First letter from the spider, second letter from the pastel. So it's pastel normal. Spider normal. And then you take second letter from the spider, first letter from the pastel, normal, pastel. First letter from the, second letter from the spider, second letter from the pastel, normal, normal. And then all you're doing now is fill in your boxes, spider, pastel, normal, spider, normal, 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 pastel, normal, 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 and same in the bottom. So to work out what you've got, you have one copy of the spider gene, one copy of the pastel gene. So that is a bumblebee, spider, pastel. That only has the one copy of the spider. This has the one copy of the pastel. This is a normal. So you can see you've got two bumblebees, two spiders, two pastels and two normals. So that's a 2 in 8 or 1 in 4 chance of hitting the bumblebee. So taking a look at something else. Here you've got spider crossed with pastel. So same again, look at the alleles. Spider, it's one copy of the spider gene, and one normal. Spider, normal. Pastel, one copy of the pastel, one copy of the normal gene. Pastel, normal. Fill out your square, and there's your odds. 1 in 4 will be normal, 1 in 4 will be spider, 1 in 4 will be pastel, and 1 in 4 will be bumblebee. So moving on to something a bit more complex, we'll do a pastel yellow belly crossed with a spider gravel. So a pastel has one copy of the pastel gene, and the other side is normal. PN. Yellow belly has one copy of the yellow belly gene, one copy of the normal. Spider has one copy of the spider gene, one copy of the normal. Gravel has gravel and normal. So this is what you're pairing. So start with this side, how to fill this out. You have P for pastel and yellow belly. So first letter of each is here. Pastel and normal here. Normal and yellow belly here. Normal and normal here. Same thing on the other side. Spider and gravel. Spider and normal. Normal and gravel. Normal and normal. And then you're just filling out your boxes. So spider, gravel, pastel, yellow belly. Just there. Spider, normal. pastel yellow belly, normal gravel, pastel yellow belly, normal, pastel yellow belly. Just continue on with the boxes, and this will give you your odds. So anything with an S in it has one copy of the spider gene. If it has a G, one copy of the gravel gene. If it has a P, has a copy of pastel. Y has a copy of yellow belly. So this would be the one you'd be aiming for. So that gives you a 1 in 16 chance of hitting odds. Another point with this one as well is if you have a here a gravel and yellow belly, they're actually located same locus on an allele. So for instance, we'll take these two spaces here. Let's say this one was yellow belly and this one was gravel. These two would be on the same allele. That's what they call an allelic animal. So you can't have a super yellow belly, super gravel. It would be a highway. So a highway has one copy of yellow belly, one copy of the gravel. And this means when it breeds, it can only pass off one or the other. It's the same with a super. If you had a super pastel, that can only pass off a copy of the pastel from this side or a copy of the pastel from this side. So that's supers and allelics. Here we have a recessive in here, so Enche and albino also have a super pastel and a leopard. So start off with the Enche. So Enche has one copy of the Enche gene, one copy of the normal gene. Albino is a visual albino, so any visuals, albinos, recessive genes, has two copies. So it has a copy of the albino on this side, a copy of the albino on this side. So it's A. Same with the super pastel. Super pastel has two copies. So pastel and pastel. Leopard only has one copy of the leopard gene and one copy of the normal. So you do the same as what we've done before. A goes here. A goes here. A goes here. A goes here. Super pastel. You have L goes here. N here. L and N here. Then it's just exactly the same. You'll fill out your boxes to correspond with the letters at the top and side. So PL, PA, that's what you're going to be aiming for as well. So this would be pastel, leopard, Enche, and one copy of the albino gene. So this would be a het albino because it's not visual. So you have 1, 2, 3, 4 out of 16, or 1 in 4. 1 in 4 chance of hitting pastel, leopard, Enche, albino. Another quick one before I go. So I would do a het pied to a het pied. So this is a recessive trait with one copy of the gene. So pied gene. If this was to only have one copy, it would not be visual pied. 2 copies is visual because it's a recessive gene. You do exactly the same one copy of the pied, one copy of the normal, copy of the pied to a copy of the normal. So in this box you'd have two P's, that's a visual pied. Pied normal would be a het pied because it has one copy of the gene. Normal pied, that's another het pied. Normal, normal. So 1 and 4 is going to be pied. Now out of the rest, you have 3 snakes that look normal. 2 of these are het pied, but you have no way of telling. So this is 1/3 of them is going to be a normal. 2/3 are going to be het pied. So that gives you, out of the normals, they are all 66% possible het pied. Until proven out. Once you're proving them out, they are 100% het pied if they breed a pied, if they make a pied in the future. Okay guys, thanks for watching. Bye.
We're lost